Hello, everyone, and welcome to the day 2 of the workshop on nutrition and metabolism. My name is Rong Tian from University of Washington, Seattle, and on behalf of my co-organizers, Jennifer Limpicott-Schwartz and Wamsi Musa, want to welcome you again to the workshop. And for those who were not here yesterday, this is a workshop on integrating nutrition and metabolism across scales. It is part of the 4D cellular physiology workshop sponsored by HHMI and the Juleen Research Campus. Yesterday, we had a fantastic day with 4 sessions with experts in the field discussing the present and the future of this field, and the discussion is extremely stimulating. And I had a great day and really enjoying the workshop. I hope you do too. So today we'll continue with 3 additional sessions on exercise, nutrition, development, and aging. And I want to encourage an audience to participate in the discussion and just write your questions in the Q&A box at the end of the presentation, and the chair of the session will read out your questions and lead the discussion. So the audience are not able to speak directly to raise their questions, but the panelists can do that. So you just need to raise your hand or put your question in the chat box. Without any further ado, we'll go to the first session on exercise. And so I will call the chair, Deb Muoio, to start.
All right, can y'all see my screen?
Yeah, looks good.
Okay, well, thank you, Rong, and really thanks to the organizers for the opportunity to participate in this really exciting workshop. I'm really pleased to be able to chair this session. Our assignment was to discuss exercise, and to be a bit more thought-provoking, we titled the session "Exercise is Medicine: Unlocking Human Potential for Physical Performance and Metabolic Wellness." The lineup here is we have 2 veterans in the field and 1 rising star, and we're going to start out with Juleen Zierath from Karolinska Institute, who's going to talk about exercise biology and give us a little history, some of the challenges and future directions. So I will stop sharing and send it off to Juleen.
All right, great. Well, it's a real honor for me to be able to be here with all you guys and present some different ideas about exercise biology. Here you go. So, all right, so I'm not sure why my slides aren't advancing, actually. Let's see if I can try that again.
Yeah, you may need to stop sharing. We're actually just seeing like a documents folder.
Okay, so we don't want to see that.
Yeah.
Let's see if we can see this one. You see that now?
You're going to have to, oh, there you go. Yeah.
All right, great.
Perfect. Yay.
All right, super. So I want to start out by just giving a little bit of history. I mean, the field of exercise physiology is not a new field. We've known since the Greeks that exercise and proper nutrition are really important for well-being. And so we can take some inspiration from that. And if we just dial back the last 100 years, there's been at least 3 Nobel Prizes that have been awarded for discoveries that are related to the field of exercise physiology. And I'm not going to go through the details of these prizes, but many of them were focused on systems physiology. So again, it's not a new field. We've learned a lot about exercise biology, but there's still quite a bit more to learn. And I think today we're looking more at the molecular resolution of how individuals respond to exercise. Exercise physiologists have really been curious about the question, how fast and how far can we go? And it still is pretty amazing to see individuals like Florence Griffith Joyner or Birgit Koschig, who can run far and fast. And for many of you, and I said this to Ron yesterday, Ron Evans, Birgit can run 5K over 25 miles faster than you probably can run in one setting. So she's an extraordinary athlete, and they have the right stuff. Dave Kostal, who is an eminent exercise physiologist, has been studying the muscle adaptation. And this is a microscopic view of Frank Shorter's muscle fibers. Frank Shorter is an American Olympian. And one can see that he's got a high percentage of these brown or dark fibers, which are the oxidative fibers. And the question is, how much of that was Frank born with, and how much of that did he train? So these elite endurance athletes not only have a great oxidative capacity, they are endowed with a high percentage of oxidative fibers, and they have a high functional capacity, a high VO2 max. They are able to perform high intensity, long duration exercise without developing a lot of lactate. So they have a high lactate threshold. They have great running economy. They're very efficient, and they can maintain their critical speed, their tempo pace for many, many miles. But we're still really interested in what makes these athletes superior. Now, what I would say, at least about exercise physiology, is that it's the ultimate integrative physiology. The adaptation to exercise training involves multiple organ systems. It's not only metabolism, but it's alterations in the central nervous system, the cardiovascular, the skin, oxygen transport, and the working muscle. And I would probably already signal that I think it's unlikely that we could ever develop one pill that's going to recapitulate all of these things that exercise does. It's a remarkable change at the whole body level and involving multiple organ systems. So I think for the purpose of this seminar, I think a lot of what we're thinking about is how can we dial in exercise and diets to optimally target many of the organs that are affected by exercise, particularly muscle, but also the cardiovascular system, the CNS, to improve aerobic capacity, overall fitness, strength, and metabolism. And there's an understanding today that it's not only diseases like diabetes that are affected by proper lifestyle and exercise, but also cancer, Parkinson's disease, many, many other diseases of aging are affected by lack of exercise and are improved to some extent by healthy living and exercise. Another pioneer in the field, John Halazzi, was one of the first to show that exercise training can enhance insulin sensitivity in muscle, and the therapeutic potential of exercise is great. Here, Halazzi showed that 7 days of exercise training had a remarkable effect to improve blood glucose control. So how can we actually understand this biology? Many of us focus on muscle. And what happens in muscle in response to exercise is that first there's a rapid increase in mRNA levels. Over time, there's an improvement or an upregulation of proteins that support a higher work capacity. And within months of training, we have an increased exercise performance and improved whole body metabolism. And when it comes to exercise, not all exercise is the same. There's high intensity strength training, but there's also aerobic exercise. And they're really defined by the amount of reps, the force that's exerted, and the training regimes that athletes undertake. We can define it into these two modalities of exercise. And we know that with strength training, that primarily leads to muscle hypertrophy. Endurance training primarily leads to boosts in mitochondrial potential in tissues like muscle. So an example of how complex is exercise is some work from Nico Pilon, who's surveyed transcriptomic studies of training and acute exercise with different modalities, strength training and endurance training. And what Nico showed was that not all exercise is the same. They lead to specific changes in the gene expression profiles. And shown on the right of this slide is that each of these modalities, either resistance or aerobic training, whether it's acute or whether it's repeated training over several weeks and months, leads to very specific gene signatures. And again, so it'd be really difficult for one pill to recapitulate all of this biology. And what I would say is that many of these genes are changing in concert. So think a little bit of a symphony. And exercise really is like a symphony. There's many, many organs, many, many genes that are changing. And I think the challenge in the field is to understand how all of that works over time and together. Here's a summary of some of what we know about exercise biology today. And again, strength training, primarily muscle hypertrophy, endurance, primarily aerobic capacity. But if you look here in the center, there's multiple transcription factors that are altered in response to exercise, and they play a role in the genomic response to training. One of the challenges in the field is to understand how all of these transcription factors kind of work temporally and what are the gene programs that they control. I think we still have many questions in the field yet to resolve. All right, so with exercise, different intensities of exercise affect a muscle cell by altering metabolism. And endurance exercise primarily leads to changes in lipid metabolism. High intensity exercise primarily leads to changes in carbohydrate metabolism. We're basically understanding that the intensity of the exercise can have an effect on fuel utilization in cells like muscle. And as exercise increases over time, there's shifts in carbohydrate and lipid oxidation. So what are these changes? How are they affected by exercise intensity? And how are they affected in trained versus untrained individuals? All right, I want to show just a couple slides, again, to illuminate some of the complexity of the organ-specific response to exercise. This is a work that was done together with Paolo Sassoni-Corset and his team, where we studied animals at 2 different times of the day. We had them exercise, and we took out multiple organs from the animals, and we did a metabolomic analysis. So this is the tissue-specific response of an animal to exercise on an organ-specific level. And one can see that there are, very, very defined effects shown here in these circus plots. If you look at sedentary versus exercise, exercise leads to a very profound disruption of homeostasis in multiple organs. And there are coordinated responses in the metabolomic profiles between different organs, with cardiovascular system, with liver, with muscle, and serum metabolites changing very profoundly. Time of day seems to also affect the way in which the organs perform metabolism. And here we're just focusing on the kind of correlated response of the metabolomic profiles between muscle and liver in sedentary animals, either at the early rest phase or sedentary animals at the early active phase. And one can see that when animals are exercising during the early active phase, during the nocturnal cycle, when they're meant to be physically active, there's a greater disruption of homeostasis, and there's a greater change in metabolites between muscle and liver, 2 main organs that control homeostatic responses to exercise. So what this shows is that multiple organs are involved. What this also shows that the time of day or the nutritional status of the animal affects the way in which organs respond to exercise. And it would suggest that, for example, if animals are exercising in the dark cycle during their day, and when they're fasted, there is a greater disruption of metabolic homeostasis. So I think where the field is going, at least when we apply this towards humans, what are the individual responses? How do individuals respond to exercise? How is this altered by the genetics that they inherit? How is this altered by the nutritional status that they have when they come into the exercise paradigm? And does disease state play a role? All right, so on the horizon, I think that there's still a lot of room to dissect mechanisms controlling both the acute response to exercise and the adaptive response to exercise training across time and across different modalities of exercise. I think that we're still in a position to understand more about the mechanisms by which different modes of exercise independently and concurrently improve metabolism and function, and in particular in different diseases. And then finally, I think one should understand that all of these approaches should be tempered with the understanding there's a lot of heterogeneity in the adaptive response of individuals to exercise. So what will be the personalized, improved metabolic control and mitigate some of the effects of different diseases? And so with that, I'm going to end here, and I hope that I've set the stage to say there's still a lot left to be learned about the adaptive response of individuals to exercise. Thanks.
All right, that was really great, Juleen. So I just, again, want to encourage everybody in the audience. This is the open Q&A session. So if you have any questions, please submit them. Try to do my best to communicate them. So I'll start off, and Juleen, I think the work you've been doing to look at the interplay between the kind of the timing, circadian influence on exercise responses is really fascinating. Can you clarify the data that you were showing? Were you sharing the acute responses that is to exercise depending on time of day, or training responses?
We've only done these studies with the acute response in mice. We've done one study in humans with a training study. And in that study, we had men with diabetes perform high intensity exercise either in the morning or afternoon. And there, when the men performed their exercise in the afternoon, there was an improvement in their glycemia. When they trained in the morning, they actually had rises in their blood glucose level, which may reflect a catecholamine response to morning training on altered counter-regulatory hormones. It may also have an effect of the diet that they had before they exercised. So I think there's a lot more to be learned about timing of exercise in healthy individuals with acute exercise and also exercise training.
Okay, I think Daniel, we have a question from you.
Yeah, thank you. What is the role of AMPK and mTOR in all these, responses?
Yeah, I mean, that's a really great question. So I'm particularly interested in oxygen sensors, HIF-1 alpha, energy sensors, AMPK, and nutrient sensors, mTOR. And I think there's still a lot, this is open. This is an open question. But AMPK, for example, is affected by the level of glycogen in the muscle. So when glycogen levels are high, AMPK activation is very low. So if you really want to have a robust effect of your AMPK, you'd want to be exercising in the fasted state. So definitely whether you're fasted or fed is going to affect the signal transduction pathways. And again, I think there's still a lot more to be learned here.
Thank you.
Okay, thank you for that question. Rong, did you have a question?
Yeah, a beautiful talk, Juleen. I was so struck by the figure you showed that the different kind of exercise change gene expression profile. There's absolutely no overlap, 0 gene shared. So how do you interpret that? Does it mean it has a parallel pathways to achieve the benefit of exercise?
Well, in some ways, I'm not surprised that there's no overlap, because if you look at the example I gave of the sprinter, she has a really different phenotype than the endurance athlete. And so if you actually want to go into the molecular mechanisms that control that outcome, I would expect at least different gene expression profiles and different transcription factors to be engaged. So perhaps it's good that there is some kind of specificity in the training adaptation and the training response. Most people don't train that way. They do something called concurrent training. So they do some endurance training and they do some weight training. And so, how is the genomic response affected with concurrent training? I think a question still to be answered. Most of us study extremes. Most of us study endurance exercise.
Okay, we have time for one more question probably. Jennifer had her hand up first. Jennifer?
Great talk, Juleen. I just am curious as to whether you've looked at the relationship of exercise, the type of food intake after or before that exercise, and the extent to which muscle is being built up, in particular mitochondria. Because the mitochondria are what build muscle, and their growth could be very much impacted by the type of food, whether it's protein, whether it's carbohydrate. I know there's a lot of athletes who really think hard about that in terms of, whether they eat carbs before they exercise, after they exercise. When do you eat a protein meal as a way to, for instance, build muscle?
So we have not done that. That's a relevant question, particularly for the athlete. I'll give one example of a study that I've been involved with my colleague, John Hawley. And we studied individuals where we controlled for the amount of carbohydrate they got during the day. But the experiment was that some of the athletes got all the carbohydrate and then did exercise and then went to bed. And then some of them got carbohydrates before they went to bed. And then the next day we studied them again. And we measured things like, PGC1 alpha, and we looked at oxidative genes, and they were much higher in the athletes that did not have the carbohydrate load before they went to bed. So you essentially had them exercise and you made them fat burn overnight because you didn't give them carbohydrate. You starved them with carbohydrates, and that potentiated the oxidative response in the muscle. And so, I mean, you know, we need certainly have more data. Your question was about strength. We haven't looked at that, but clearly substrates on board can affect the outcome, whether it's oxidative or glycolytic potential of a muscle cell.
Okay, so great discussion. I want to say there are a few questions in the Q&A box, and I will try to work those in the general discussion. A lot of good points there. But I'd like to move on now to Ron Evans from the Salk Institute. Ron is going to talk about breaking barriers in human fitness and health span. You might tell us a little bit about the exercise pill. Ron, you're muted.
Sorry, can you see this? Do you see my screen?
Yeah.
Okay, good. All right. So what I'm going to do is, and thanks to Juleen for giving a great intro.
Actually, Ron, have you shared your screen yet? Because I don't think we're seeing it.
Okay. Let me see. Sorry, let's see.
Yeah, we see it.
Okay.
It's close. Just put it in full screen.
Okay.
There you go.
Okay. All right. Thanks, Juleen, for the great intro and getting this section jump started. Literally jump started. We'll tell you. And I'm going to continue with the theme of exercise physiology more at the molecular genetic level as well, and raise a question about the potential for something that I call superphysiology. And the slide here just shows the morphing of the DNA double helix into a tape measure, which is your waist, and the increase of lipid and other components as we gain weight. And there's an intimate relationship between the physiologic programs that are encoded here and what's happening as we take nutrients in. And exercise or not. So the next slide, just to sort of develop the idea here, is looking at the nuclear receptor superfamily. They work by controlling nuclear networks, by integrating physiology, and by integrating behavior and impacting on disease. And I showed in the context of a clock, that's time, and then sort of the background, which is space. So space and time. If you look at nuclear receptors very briefly, we've clustered them based on what receptors like to be next to each other or co-express. This is GR, LXR, and PPR, sugar, cholesterol, and fat. Cluster, they're not related sequence, but they are related into what they do. The other is up in this pattern up here, reverb, alpha or beta, and ROR and ERR. So these are circadian regulators here. And what groups with circadian regulators are oxidative nuclear receptors, as well as thyroid hormone receptor, basal metabolic rate. So this suggests to me that circadian rhythm is really metabolic rhythm. And that's an important point from the genetic concept. So when we think of systems physiology, it's a process that occurs, as I mentioned, in space and time. The question is, what's the molecular architecture of a physiologic pathway? The different physiologic pathways have common genetic underpinnings, and how are these physiologic pathways integrated? So from Darwin, he coined survival of the fittest. We can kind of look at that. We're not too far after his 200th anniversary here. So he discussed and originated the idea of origin of species. but no one ever really asks about the origin of physiology. And I think that this is an important question, because if you understand the origins of something, then the logic of how that works and the mechanisms by which it arose genetically is going to be very revealing. Another key feature that we should talk about with this is the concept of homeostasis, which is the maintenance of the environment, whether it's sugar, salt, calcium. This was by Claude Bernard, called The Basis of a Free and Independent Life. This is what he considered in his book, The Principles of Physiology, and the study of organic functions, including endocrinology, kinesiology, so it's exercise, enzymology, nutrition. He was already thinking very far ahead in what physiologic homeostasis might be. Jump ahead to the nuclear receptors, which I show here very briefly, and the nature of the superfamily. Virtually all of the hormones, the initial hormones in the superfamily, vitamin A, which was the first vitamin isolated, vitamin D, the fourth one, these came 1 year apart, thyroid hormone, basal metabolic rate. These were all discovered within a year of each other. Then came these steroids, glucocorticoids, and mineralocorticoid. And each of these were tremendous advances, controlling sugar. And the last key molecule in this was the discovery that bile acids and vitamin D were related to each other. That was a surprise, and that was a Nobel Prize by itself. And virtually each one of these were Nobel Prizes because they were so striking. The chemical structure of these hormones are very different, but the target turns out to be the nuclear receptors, which are shown down here. And that means that there's a shared mechanism between all of these, what were individually isolated hormones. This stayed basically in this position for another 75 years or so. And with the isolation R cloning and the structure of the first nuclear receptors, we realized by homology that there were other receptors potentially in the genome. And that led to the discovery of orphan receptors, which some of them are shown here. And with orphan receptors, we went from having 9 signaling pathways to another 38 unrecognized metabolic pathways and signaling pathways that were completely unknown. And so that opened up from those 9 to another 38, a whole new opportunity and an arena to explore now an expanded version of the underlying physiologic mechanisms. So I'm going to talk about briefly 2 oxidative receptors, PPAR delta, and a little bit about ERR alpha. These networks, physiologic networks are genetic networks. Physiologic mechanisms are basically from the gene to, let's say, to the muscle. So it begins with genetic circuits. Some traits are intrinsic and some are innate. Others are adaptive. If you look at this, spotted hyena is very endurance. Leopard is speed. If you're a tailed godwit, you can fly 6,500 miles straight from Alaska to Australia or New Zealand. You can't train. So these are genetically encoded, whether you are fast or have endurance. And humans really have a combination of both. We are really adaptive. We can run fast. We're one of the greatest endurance animals. So this is interesting in deconstructing what is our genetics allow. This just shows the balance between energy storage and energy burning. And I'm going to just mention briefly a little bit more about PPAR delta. This is the PPAR family in terms of energy consumption, as opposed to PPAR gamma in the adipose depot, which stores energy. And so, as Juleen mentioned, mechanical exercise or physical exercise is depending on how you do it, a oxidative or a anaerobic type of program. In terms of PPR delta, in terms of PPR delta, the idea of trying to activate this receptor alone using small molecule drugs or genetics was one of the areas that we began exploring about 15 years ago, actually. I'm just going to show you not the whole film of this, but if you want to look at mice that are running, and this mouse is not running very well, what we see is a mouse that has not received the drug. These are both sedentary mice, and one that did receive the drug, and these mice have been pitted against each other in this treadmill challenge. And I'm just going to kind of skip ahead a little bit. And what you can see is while they're keeping pace for a while, the mouse on the left has now fatigued. That's the one that did not get the drug, whereas this untrained mouse keeps going for another hour. And so that began our interest in how is the drug able to do that? I'll show you a little bit more. This is newer data about the impact of the PPR delta drug. We're calling it MA here. Control mice run. Looking at VO2 max, you can see how the drug increases VO2 max significantly without any exercise. If you train, VO2 max on its own goes up, and it doesn't increase with training. It doesn't change after that in response to drugs. So I think that's very interesting. If you look at running time in the same thing as endurance, a control mouse can run 176 minutes on average. If you give them the PPR delta drug, 300 minutes without training. Training alone gives you 256 minutes, but the combination of training and the delta drug gives you 464. And this mouse up here was running just short of 10 hours. That's a huge amount of time for a mouse to continuously run. And it takes a long time. You have to watch each mouse individually. And so this is a really amazing distance for a mouse to run. And the question is can you create something that we might call super physiology? And in terms of these different parameters, just alone giving control, training, the drug, or the combination has a big impact. And so we're very interested in how that happens because we're targeting one single pathway, which is the PPR delta receptor. Juleen showed you this pathway. It's her pathway. She described it. And I'm just going to modify it with some of the ideas of John Halsey, as she mentioned, that there is a substrate shift that occurs after the PGC1 alpha component that is mediated in part by ERR alpha, the very oxidative receptor, and then PPR delta. And that leads to increase in mitochondrial number and increase in metabolic shift to burn lipid over fat. I'm going to kind of skip ahead to basically see how the drug is impacting this pathway. And just to quickly end, if you look at physiologic benefits of exercise that were shown here and Juleen mentioned, from endurance exercise, you get these components, and basically with the PPR delta drug, you get the same components that are being activated. That doesn't mean that one pill is the same as mechanical exercise. It is not. But interestingly, targeting one single component, which is at the core of exercise, has a major impact. And that opens up the door not to just exercise physiology, but more general, can physiologic pathways be gamed based on our understanding of their underlying genetics? And how can that help? And what do we learn from that? So I'll stop there and take some questions.
All right, thanks, Ron. We have a couple minutes for questions. I don't see anything in the in the Q&A box just yet. So let me ask, Juleen talked about, the acute versus the chronic or adaptive responses to exercise. And I'm wondering if you've had a chance to compare the PPAR delta compound and how it initiates an acute response every time the animal is dosed versus the chronic response, and compare that to acute versus chronic exercise. How much overlap is there, and how much distinct activation is there of some of these networks?
That's a great question, and it's what we I don't know if you say we train, but we give the mice 8 weeks of the drug, and they're run depending on if they're being trained or not. If they're being trained, they run an hour a day to put them in shape, or we don't run them at all. So those are going from trained to not trained. And that's when you get that significant approximate, just not quite two full difference in that time. And depending on the dose of the PPAR delta, you can increase that pretty dramatically. But in that 8-week time frame, you get to the peak maximum that can be achieved under any circumstance in the combination with training and the drug. You get to a very high level very fast. It can be compared to in the acute setting, you're really dealing with intrinsic features that are basically based on the animal's activity or not. Some animals are actually pretty active and others are relatively sedentary. And if you take strains of mice that like to run in the running wheel, they get a fair amount of good energy. They burn a lot of energy and they train themselves. And so, so in the acute state, you kind of have the basal part of a physiologic mechanism, and in the activated state, you have this sort of component of the program, in our case, where you can accelerate or jumpstart that program that's there, but it is adaptable because the human program is adaptable. You can actually push that to where you want it, and that gives all sorts of benefits.
All right, well, thank you. I think we'll talk more about that in the discussion session. There's not any questions from the audience that I see, so just stay on time. I think we'll move forward. And it's a real pleasure to introduce Dongyin Guan from the University of Pennsylvania, who I believe is going to talk about circadian control of metabolism.
Yeah. First, thanks the organizer pick up me to give a talk here. It's a really great pleasure. Can you see my screen? Can you hear me? Oh.
Yep, looks good.
Oh, looks good. Okay, great. So I'm not the expert in the exercise, so I pick up this picture, mimic the exercise. But my major focus in the liver. But I think a lot of research strategy and also like inter-organ crosstalk can apply to exercise. I will start my talk from this book. Actually, Ron just mentioned from Charles Darwin. There's one paragraph said it is not the most intellectual or most strongest organs species to survive. Actually, the one able to best adapt and adjust to the changing environment. Actually, we face the changing environment every day due to the Earth's self-rotation. That's the reason we have in our body and same to other organs, the gene, hormone, even we discuss a lot metabolites, they have circadian rhythms. With the effort from many years, from many labs, for many years, now we know there are several transmission activator and repressor. They form this interlocked feedback to control circadian rhythm. If you do loss function study, you will find in different tissues, disrupt circadian rhythm cause a lot of metabolic disorders. For example, in the muscle, they cause, affect mitochondrial function, as mentioned by Ron also. Also, in the liver, they also affect lipids and also glucose homeostasis, and more than that. when joining the lab as a postdoctor, I'm pretty interested in several key questions in the field, still not answered yet. The first question is how this internal clock machine responds to the nutrient stress. Second question is what's the tissue-specific regulator? The core clock genes that I just mentioned in last slides, which exist in every cells. But if you check the circadian rhythm in different organs, they are differently. So they have to some tissue-specific regulator coordinate or independent regulate circadian rhythm. So for this talk, my central hypothesis is a non-canonical clock regulator plays an important role in this regulation. So the method I applied is called functional rhythmic epigenomics. So I use a system called data-induced obesity because epigenetic is the interface between environmental clues and also internal homeostasis. So basically DNA sequence do not change, but you have different stimuli. You can change the DNA or chromatin architectures, then change the enhancer driving the gene expression. Once we know a bunch of gene enhancer be changed, so we can predict what transfer factor bind to this specific motif. With this, we can do this kind of a function study because you make a prediction based on genome-wide unbiased study. Find the hits, then you do those function study. And we also know, just like people data or other factors, they also have circadian rhythm. So if we know what's the best time to activate them, then maybe we can also maximize the drug effect or minimize side effect. They call the chronopharmacology. Or we also know different time to eat also can affect lots of circadian rhythm. That means what's time to eat. And also Juleen just mentioned maybe what's the time to exercise. That's another chronotherapy approach. So the approach I used here called the global run-on sequence basically is unbiased way to determine the enhancer activity and gene expression in the same data set. So as you can show here, this is the normal mouse activity similar to the behavior. This in the dark, they are more circadian and less active in the night. But in this kind of half-light condition, the circadian rhythmicity is kind of attenuated. So this basically this mice is circadian disrupted mice model. Not surprised, we observe thousands of genes and enhance lots of circadian rhythm. Interestingly, we also observe another group of genes. You can see here, they are not that circadian, but they become circadian in this circadian behavior disrupted mice, similar to the enhancer activity. So we can correlate it with the enhancer with these genes. Then we can do motif analysis. They can predict what motif regulates this enhancer and downstream genes. With the motif analysis, we find two top motifs. One is Ron just mentioned PPAR alpha, one nuclear receptor. This also Joe mentioned this is related to fatty acid oxidation, burning the fats. Interesting, we also find another totally opposite pathway is lipid synthesis. They are synchronized together at 5 p.m. and have maximal transcription activity. Will have lowest transcription activity in the antiphase in the morning, 5 a.m. So whether this kind of bioinformatic prediction is correct or not. So we did the validation. This gene expression, they gain circadian similar to the target gene. Working with Josh lab, Josh Obrowski lab, he just gave a fantastic talk yesterday. Did the in vivo tracing, determine the in vivo lipogenesis and also oxidation. You can see in the oxidative, in the diet-induced obesity mice, they are synchronized at the same time of the day. So we did a loss function study to check the causal effect, and the answer is true. So to make a long story short, so make a summary for this story. Actually, it's very interesting because in a healthy liver, lipid synthesis usually inhibit oxidation. That's evolutionary makes sense because you want to eat something, store the energy. You don't want to burn it at the same time. But in this overnutrition environment, these two lipids pathways synchronize together, synthesis and oxidize together. I think is a kind of response to the overnutrition environment. Actually, when active PPAR alpha at 5 p.m., we can kind of increase the oxidation rate compared with 5 a.m., show some chronotherapy potential here. In this story, and also in the introduction, I told you this core clock regulator are very important to regulate circadian rhythm. Then we check this core clock genes in this normal chow and diet-induced obesity mice. you can see there's not huge difference. The big difference may be RR gamma, but not as huge as PPAR alpha, the SRBP, which I just show you. The question is, what's the function of these clock genes in the liver, and how liver clock is regulated? To answer this question, we use this AV cream to delete reverb alpha and beta in the adult mice. I also aware Ron have a paper use the albumin cream knock out the reverb alpha and beta. And we do observe some similar effect. First, we observe knockout reverb alpha and beta. The knockout efficiency is pretty good, it dramatically decrease. There's some signal at peak expression. Later I will show you data. This is from non-hepatocyte. Because this system, the knockout is specifically to the hepatocyte, single cell type. More importantly, you can see this target gene and other cockroach genes. Their amplitude decrease from 30 to 300 decrease to less than 1.5 fold. Basically, this liver is circadian cockroach gene disrupted liver model. So what's going on with the circadian attribute genes? Consistent with Ron's paper and our previous GND paper, we do observe lots of genes circadian rhythm disrupted from control to knockout. They don't have circadian rhythm. Actually, interestingly, we also observe another two group of genes. The circadian rhythm still retained, although amplitude may be decreased or increased, but generally they don't change too much. And even some, they are originally not circadian, they gain circadian rhythm. Actually, we did this kind of changing the feeding behavior, basically force them just provide the food in the daytime, just force them to eat in the phase, later phase. Actually, this kind of circadian genes retained is disrupted. So feeding fasting cycle also contributes to this circadian rhythm in addition or collaborative with the kind of endogenous clock. But this is not today's focus. Today, I want to kind of in the last part, I want to zoom in from the tissue level to cellular level. Yesterday, we talked about zonation and single cell resolution. And also yesterday, we in the liver section, we talk about zonation, different cell type in the liver, which means there are different clock in different cell type. So I would like to know whether there's prefer clock communication between different cell types. That could show some synchronized behavior to perform the overall tissue level physiology function. But to do this, I did this kind of single nucleus sequence because I did not do single cell because we know knockout the clock genes can cause some fatty liver and they cannot pass to 10x genomics and also cause some bias. In this approach, we can capture most of cell type in the liver. Moreover, when we disrupt this clock specifically in the hepatocyte, we can see the different gene change in the non-hepatocyte, suggest clock in this hepatocyte can affect clock in non-hepatocyte. Just a control here, just in case I knock out reverb in this hepatocyte can affect gene expression in non-hepatocyte. Here is the control. You can see the knockout efficiency in the hepatocyte is pretty good. This is hepatocyte, and target gene is de-repressed or lost circadian rhythm. But in endothelial cell, calf cells example, their circadian rhythm don't change for the cockroach genes, and the knockout merely change here. How about the circadian attribute genes? I show you one example. You can see the circadian rhythm is a lost circadian rhythm or gain circadian rhythm in endothelial cells, same to the calf cells, same to the endothelial enhanced landscape. This is kind of a gene expression. We also work with Josh's lab to work about the metabolites. This is gene expression in this GSH glutathione diphosphate pathway. The gene expression lost circadian rhythm. There's more genes. This is one example. This metabolites also lost circadian rhythm in endothelial cell where we disrupt clock in hepatocyte. We can integrate gene expression and also metabolites, define the different pathway be affected. This is the glutathione metabolism and some lipids metabolism. And we can do more later. But due to time limitation, I will directly go to the conclusion part. So in the first story, I show you in the diet-induced obesity mice, there's a profound circadian remodeling, but cockroach genes don't change too much. In the second story, even we disrupt these cockroach genes, there's some genes circadian rhythm still retained. In non-hepatocyte, the clock genes don't change. They also have reprogramming. Actually, interestingly, we published this paper at the same time. Juleen also published another paper in the human. they said they find also time-restricted feeding changes a lot of metabolized circadian rhythm, but also do not disrupt these cochlear genes. All this consistent evidence indicate there is kind of tissue-specific or non-chronological regulator playing important role in this circadian rhythm. So this non-chronological regulator could be tissue-specific or physiological context, like diet-induced. So I think this could be another drug package, for chronotherapy or determine what's the best time to eat, or in this section, what's the best time to exercise. With that, I thank a lot of people involved in this project. And this picture just take last week, since I will leave Mitch's lab, go to Baylor, start my own lab. Thank you very much for your time, and I would like to take questions.
Right, thank you, Diane. That was it's really beautiful work. And so let me start. I'm going to try make an attempt at integrating your talk and Juleen's talk. And so my impression is that a major driver in terms of how the system is responding to the clocks is because the clocks and the circadian programs are regulating nutrient metabolism, and in turn, nutrient metabolism is affecting form and function. And so A, am I in the ballpark there? And B, so translating that to Juleen's talk, would you suspect that the reason why exercise training has different outcomes depending on time of day is because of the influence of the clock, or the influence of the nutritional stress that then feeds back on the clock?
Yeah, so my quick answer, I think the key point is synchronize. So when you exercise, then you active or repress certain pathway, can they kind of synchronize or fitness, use the word wrong, that can fit to the internal clock pace. So if I'm not sure, in my impression, maybe good time to exercise in the afternoon or not, I'm not exactly sure. But I think when you have this kind of oxidative or AMP activation, they can fit the endogenous clock. I think that could be the key. Yeah, and whether it's exercise affect the metabolism or metabolism affect exercise, I think that is mutual. That's my quick answer. I'm not sure I answer your question. Yeah, or other people to provide insights. Yeah.
Any other questions from the panel here? Now, one other question that came to mind is, the rhythmicity of the gene expression signatures is really interesting and compelling. Do you know at this point how well or not the transcriptome aligns with the proteome?
That's a great question. Actually, let me very quickly share that. So due to time limitation, I did not show this slides, and I think this is the kind of slides last slides show for this workshop. So I think there are different layers of the regulation. One is gene transcription, and also you just mentioned the proteomic translation and the protein decay, and also metabolize. I think that's multiple ways. But for the how overlap of them, actually we do not find the huge overlap. That means that in each step there is additional regulatory, the control decay, control synchronize, and also for the metabolize, control also transport. I think that could be another direction or future direction. We need a creative view and tools to study this synchronize, either in cell-cell communication, either in intra-organ, and also go back to your question, different regulatory level, transcription, translation, post-translation regulation, and then metabolize. Yeah, thank you.
Can I just answer part of that question?
Sure.
The diet does impact exercise. And if you put mice on a high-fat diet, for every gram they gain, they move a little bit less. And as they become obese, basically they move very little. And this is important because in our society, for heavy people, we say that they're lazy and they're not getting exercise, and the problem is their fault. And it's kind of a way of trying to shame people into losing weight. But in fact, and if you look at the mouse, they're not lazy. It's intrinsic feature of weight gain that has a central effect on exercise. And so I think this is a dynamic way of looking at why diet in part can be dangerous and cause all sorts of problems if it's a high-fat diet and if you don't get exercise. And so the high fat and the fitness side are two opposing features. If you burn energy, you're going to be much healthier. And if you store it for a long time, you're not going to be so healthy. So there is kind of this direct interaction.
All right, well, great presentation, Dongyin, and from everyone. I think just to try to stay on time, we'll move into the general discussion section. And so let me start by just saying, at least in my mind, one of the, I think, the long-term goals of exercise research is, is trying to understand mechanisms so that we can enhance a human health span. And so, in the field, there's debates about, well, whether or not we should strongly consider exercise mimetics. Juleen made the point that it could be a little naive of the field to think that we're going to recapitulate all of the molecular networking that's activated by exercise. On the other hand, then Ron makes a strong case that at least in the mouse models that you can mimic some of the the really beneficial effects of exercise. So so maybe we could have kind of start in that areas. What do we view as the ultimate goal of exercise research going forward? And then a question that came from the audience was, how do we actually move what we're learning in basic biology, whether it's the impact of nutrition or timing of day or some drugs that tickle the same pathways? How do we move that into the clinic where we could actually have, it could benefit patients? So, Juleen, maybe start there. I know it's complex, right? It's super complicated.
It is complex. Well, listen, I'd argue we have a few exercise mimetics. I mean, I would say a statin is probably an exercise mimetic. I mean, it lowers LDL cholesterol, and exercise does that. So there's definitely room for things that mimic certain effects of exercise for people who need help in those areas. I mean, ultimately, I guess as an exercise physiologist, I'd like to see people really exercise. And if we can try to instill that in people in their lifestyle from all, the span of life, that would be a great thing. So I would be a proponent of saying it's really important to try to have people move and understand the benefits of physical activity and diet. And we're seeing this now with a lot of the devices that people have. Everybody has one of these watches. People are really paying attention to that. So it's answering part of your question. There's a lot there, Debbie.
Yeah. Okay, well,
I would say one other thing about that, which is the nature of real exercise, as we would say, is really pro-health because it activates an oxidative program. And that's what PPR delta is doing as well. And I think that the target of PPR delta specifically and of exercise are mitochondria. Mitochondria are kind of passive players. They take their cue from what's happening. They're being controlled by, let's say, PGC1 alpha because there's a exercise trigger, and they'll become more oxidative. And so the mitochondria are really playing to the needs of the cell or the organ that they're in. But the target is the mitochondria, and that's one of the big targets for PPR delta. However, PPR delta does not promote mitochondrial proliferation. It promotes specifically oxidative metabolism and reduces the use of glucose in favor of burning lipid. And so there's a very logical feature for how you get that oxidative metabolism ultimately linked to burning fat, ultimately, which is what endurance exercise is. And so there's a really strong underpinning between the links between the diet, the target, which is the mitochondria, and how you get to that mitochondria, and how the mitochondria, I'm not saying interprets it because it doesn't have a brain to interpret, but it's designed to be the servant of the metabolic needs of the particular organism.
Okay. So we have a couple of panelists who know a lot about mitochondria. So
Don't forget resistance training and strength. That's probably more important for the average person is to maintain strength and balance.
Yes, yep.
Okay, so Vamsi, you had your hand up.
So I have one question for any of the panelists, but I think maybe it would probably be most appropriate for Juleen, perhaps. So we always hear about the health benefits of exercise. I'm just wondering, are there any health conditions that are actually worsened by exercise? And I'm not talking about injury, musculoskeletal injuries, but
I can't think of anything where there'd be a deleterious effect of exercise. And again, exercise, we're just using the word exercise. Walking is exercise. Walking up steps is exercise. You know, I mean, it might not be training for the Olympics, but all of these things, physical activity, we could talk about that as well. Physical activity. So I would say that movement, we're made to move. So I can't imagine a state where we're better off by not doing anything. And maybe Debbie, I don't know if you have an opinion about that, but I think we're made to move.
Yep.
Well, agreed. And I think, the beauty of biology is somehow the muscle and the muscle mitochondria, they sense what's needed, right, in response to the activity, and somehow it's balanced. And, a question or a consideration when targeting a single pathway is, are you building, your mitochondrial reticulum, which ultimately then consumes more space in the muscle, reducing the size of the myofibrils, and then potentially compromising strength if that's not balanced the same way that biology balances. So, so yeah, definitely a lot of complexities there. Let me go to Bruce because he's had his hand up for a bit.
Well, let me make a quick comment about the health benefits. There are examples of where exercise is not beneficial, is deleterious, and there's a version of anorexia nervosa where people become addicted to exercise, and it can even be life-threatening. That's an extreme example, but it is a real example, and it has become increasingly prevalent among adolescents, and it is something to be taken seriously. So yes, there are examples of the deleterious effects of exercise. But more to the point, I wanted to address Deb's question, which is how we can think about exercise and how we go forward. And I'm going to touch on this in my own talk in a little while, but just to say, exercise is also a platform to discovery molecules that potentially have beneficial effects, both metabolites, polypeptides, who knows what else. But my own approach to exercise has been to sort of try to find out what's downstream of the exercise. I'm very glad people look at these other aspects, but I'm very interested in what is downstream of the exercise that talks to the brain, that talks to the liver, that talks to the adipose tissues. And so we all have our own niche in this field if we're doing good science, and we need all of the above. But I am personally enamored, excited about the idea that something in exercise talks to the brain, and it has effects on cognition. Something talks to the liver and deals with the liver accumulation of fat. So I think there's tremendous potential in using exercise as a platform for the discovery of other therapeutically important molecules.
Yeah, I mean, Bruce, that's really the integrated physiology. I mean, it's really a miracle, actually, how all of these organs can kind of work in concert. And that's what I said, also the symphony. How do these organs all play together to produce outcomes?
Jennifer, your hand is up there.
Great. So this is to continue the discussion about with exercise, how do you change, what are the changes that correlate with this? And I'm particularly intrigued by Ron's work with the drug that's mimicking PPR delta. And I just wanted to ask you, Ron, so you say that the mitochondria don't proliferate under these conditions, but does the muscle of these mice get bigger? Do you see brown fat? And is there overall temperature going up?
We don't see
driving more lipid. If you're driving more fat consumption, you're either, I mean, is it going out as ATP to generate, you know, the actual muscle contractions, or is it generating heat in some way? Or is heat a byproduct?
Well, it increases the P period delta. It does increase heat. It does not promote, and it does that because it's promoting oxidative metabolism. So you're burning lipid, which creates heat. You know, the product of any kind of exercise is heat in the end. And so mice are just getting, obese mice just getting the P period delta drug to lose weight. And you can see increased thermogenesis. You see increased browning as well, particularly in the white adipose depot. In the context of the mitochondria, while it does not promote mitochondrial proliferation basically at all, exercise does. And even mild exercise does that. That's why when we mix training with the P period delta drug, you get a boost. So you're then having an oxidative program that is being enhanced by something that's stimulating mitochondrial numbers. And so it's, you know, so you have those two things coming together. Again, it ultimately focuses on trying to get the mitochondria to do what you want for the kind of exercise that you're doing. And so if you combine the two, then you get this sort of jumpstart or accelerated progress in converting from, let's say, a sedentary state to active or more oxidative state.
And if those animals are fed a protein-rich diet or was ketogenic, they might run into some big problems, right? Because they won't be able to make fat. And if they're burning fat, it would seem like they need to have a carbohydrate-rich diet to continually to build the fat that they're going to be.
Yeah, I mean, I have to think about that, because if you see the best athletes in terms of endurance, they're like the thinnest people that you ever see. I mean, they don't need a lot of fat because their bodies are really tuned to oxidative metabolism. They're kind of overtraining in that, it's not having too little fat in that context.
Fat that's going to be burned, basically.
Sorry? Yeah.
They need carbohydrate big time.
Oh yeah. Oh, no, yeah. You need carbohydrate. Yeah. But the point is that when you're burning fat, you always have sugar in your blood because you're not burning down that sugar. And that's also important for central function. So one of the things that endurance runners do is they burn fat. And by doing that, they don't burn sugar. If you do burn sugar, then you can hit the wall. You get your sugar levels too low. And when that happens, that's what you can't do anything. You become overwhelmed and you basically fall on the track.
Let me jump in here. Most athletes, when they're competing, and it's particularly if they're running the marathon, they ingest fuels. Yeah, so it's not just water. They'll take carbohydrate. They'll use gels. So the endurance athlete does nourish themselves while they're performing work. So they really generally don't get completely depleted. That's the point of their training. And it's a point of their training regime is also to use nutrition as a part of that performance.
Great, thanks.
So Ron, you made the point that a major component of the mitochondrial remodeling is really driving the fat oxidation machinery. So so one of the questions we have from the audience is that, are you then implying that any stimulus, whether it's pharmacological or nutritional, or anything that enhances fat oxidation should in turn enhance endurance and have positive impact on health without driving mitochondrial biogenesis?
Oh, without driving mitochondria. Well, yes, we show pretty definitively that without any exercise, the drug does confer. You can sort of disconnect certain components of the complete exercise pathway and get very good oxidative function, which is beneficial, as we've shown, to cardiovascular disease, to liver disease, and to diabetes, particularly insulin-resistant based diabetes. And so you don't have to be physically active in that context to get a benefit. It's much better, as you saw from what we showed, that even if you get mild exercise, as Juleen said, walking, that by itself is actually reasonably effective. If you combine that mild exercise with the PPAR delta pathway being activated, you get really a much more synergistic effect. So in that context, you could think of that as a therapeutic way to advance that program, particularly since 2/3 of this country are basically obese, and many of them get very little exercise. You can't tell people to go out and exercise. That's been happening. You've been doing that for decades and decades and decades. And we just keep getting heavier and heavier and heavier. And so you do have to think of the burden, financial burden of people getting overweight and the cost of that, and whether if you can intervene in some way to synergize the small amount of exercise to increase their oxidative state, would that be beneficial? I think it's definitely something worth considering.
All right, well, to stay on time, I think we'll wrap up the discussion section. This is again, real provocative discussion, and I look forward to more thought about how Genelia could develop new tools that might help the field move forward at a faster pace later on this afternoon. So thanks all for your attention, and we'll turn that over to Ron.
So actually, we're going to take a quick break right now. We will break for about 5-6 minutes and resume at 12:20 Eastern.